Good evening, everyone. You can call me Michael. And first, I'd like to thank the organizers for giving me the opportunity to present some data I've obtained working as a postdoc in the addiction research group led by Stephen Laviolette. In the lab, one of our main objectives is to better understand which brain circuits and molecules underlie the effect of cannabinoid compounds on cognition and emotions using rats as animal models. And in this context, my attention is particularly focused on cannabinoid receptors in a specific brain area called the hippocampus, and on how modulation of these receptors can impact memory formation and social interaction in rats. Cannabinoid receptors are a class of cell membrane receptors. There are currently two known subtypes called CB1 and CB2. The CB1 subtype is expressed mainly in the central nervous system, whereas the CB2 subtype is expressed mainly in peripheral tissues. Both receptors can be activated by three major groups of ligands: by the endocannabinoids produced by the mammalian body, by phytocannabinoids produced by plants such as marijuana, and by synthetic cannabinoids such as Win55. Both receptors can be in the central nervous system. Activation of CB1 receptors by cannabinoid compounds has been shown to alter emotional perception and social interactions both in humans and rats. And it is also known that it can affect memory processes by increasing the learning of non-salient information. And we are currently trying to understand which brain circuit is responsible for these effects. One brain region strongly linked to memory and emotional processes is the hippocampus. Both in humans and rats, the hippocampus is a bilateral structure located in the medial temporal lobe. In humans, hippocampal dysfunctions are associated with psychological disorders such as schizophrenia, in which cognitive functions and social interactions are strongly altered. It is also known that in rodents, lesion of the hippocampus can alter memory and emotional processes. The hippocampus contains a high density of CB1 receptors, whose activation significantly increased hippocampal principal neurons activity. Moreover, activation of the ventral part of the hippocampus can activate dopaminergic cells located in the ventral tegmental area. Once activated, this population of neurons release dopamine, a neuromodulator involving several cognitive processes, such as the detection of salient information. This control of dopaminergic cells by the ventral hippocampus is not direct, but is made possible via modulation of neurons' activity in the nucleus accumbens, a brain region involved in the integration and transformation of information into adapted behaviors. Based on this data, and because activation of CB1 receptors in the ventral hippocampus can increase hippocampal neurons activity, our hypothesis was that activation of ventral hippocampal cannabinoid receptors would increase dopaminergic cells activity and would impact memory formation and social interactions in rats. Thus, the first step of my experiments was to study the consequences of an infusion of a CB1 agonist into the ventral hippocampus on dopaminergic cells activity. And to do this, I performed recordings in rats under anesthesia, in which I lowered a syringe into the ventral hippocampus in order to infuse Win55, a CB1 agonist. At the same time, an electrode was lowered into the ventral tegmental area, allowing me to record the electrophysiological activity of dopaminergic neurons. Typically, the experiment started by the recording of the baseline activity of a single neurons before a single infusion of Win55. Then, the activity after the infusion was recorded and compared to the baseline activity. I'm sure you want to see the data. 20 neurons were recorded in this experiment, and their firing frequency expressed as the average, and their activity is expressed as the average frequency before and after the infusion of Wnt55 into the ventral hippocampus. The yellow star indicates that right after the infusion of Wnt55 into the ventral hippocampus, the dopaminergic cells activity significantly increased. And in terms of number of neurons, 70% of the recorded cells significantly increased their activity right after the infusion. With this data, I had an evidence that activation of CB1 receptors in the ventral hippocampus significantly increased dopaminergic cells activity. In a second experiment, I tested whether the activation of CB1 receptors in the ventral hippocampus would promote the learning of a normally non-salient information. To answer this question, I used a conditioned place preference protocol. And in our protocol, the rat is taught to associate a particular environment, that is a white or a black box, with an injection of either a saline solution or a subthreshold dose of morphine. Development of a place preference is then assessed during a test day. And during this test, the rat is placed in the gray area located between each boxes, and the time spent in each compartment is recorded during 10 minutes. If a rat develops a place preference, he will spend more time in the box previously associated with the morphine injection. But if a rat doesn't develop a place preference, he will spend the same amount of time in each boxes, and this is what we obtain by using a subthreshold dose of morphine. This is the design of the experiment. Wnt55 was microinfused into the ventral hippocampus through a cannula implanted in the rat brains. And this microinfusion was performed before each acquisition session. Then, rats were tested for their preference 3 days after the end of the acquisition. And their performance were compared to that of a control group, which received a vehicle solution into the ventral hippocampus before each acquisition session. This histogram represents the time spent by the control group and the rat treated with Wnt55 into the ventral hippocampus in the saline box in white and in the morphine box in orange. As you can see, the control group spent the same amount of time in each boxes. Conversely, rats treated with Wnt55 into the ventral hippocampus spent significantly more time in the morphine box. Thus, this group of rats developed a place preference. I further tested whether a co-infusion of Wnt55 and a selective CB1 antagonist would block the place preference induced by Wnt55 alone. And as you can see, it did. And these groups spent the same amount of time in each boxes, like the control group. Thus, activation of CB1 receptors in the ventral hippocampus can induce the learning of a normally non-salient information. Then, I tested rats' natural sociability. In this test, the rat is placed in the middle chamber of a 3-compartment open access area. A second rat, which has never been in contact with the first subject, is kept in a small wire cage in one of the 2 lateral chambers. Here, the time spent in one of the 2 lateral chambers is recorded and compared. This test is based on the natural tendency for the rat to spend more time with its fellows than in an empty area. But is it the case if we infuse Wnt55 into the ventral hippocampus before the start of the experiment? This graph represents the time spent by the control group and the Wnt55 group in the empty box in white or in the stranger box in orange. As expected, the control group spent significantly more time with its fellow. On the opposite, rats treated with Wnt55 into the ventral hippocampus did not show a significant preference for the box containing the stranger. Co-infusion of a selective CB1 agonist with Wnt55 into the ventral hippocampus reversed the effect, block the effect of Wnt55 alone. Thus, activation of CB1 receptors in the ventral hippocampus can abolish rats' natural sociability. Based on previous data showing that cannabinoid compounds can alter memory, emotional perception, our working hypothesis was that activation of cannabinoid receptors in the ventral hippocampus would impact memory formation, social interaction, and dopaminergic cells activity, a neuronal population involved in these later functions. By using a combination of approaches, including electrophysiological recordings, selective pharmacological treatments, and behavioral tests in rats, we found that activation of CB1 receptors in the ventral hippocampus can influence dopaminergic cells by increasing their activity. It can also influence memory by inducing the learning of non-salient events. And it can impact social interactions by altering natural sociability. There are more than 60 million cannabis smokers around the world, and 40% of Canadian adolescents have tried cannabis at least once, maybe you. Moreover, cannabis intake during adolescence is considered to be a high-risk factor in the etiology of schizophrenia. So I hope that the research we perform every day in the addiction research group here in London, Ontario, will help to understand what's happening in the brain of these guys. Let me finish by thanking our funding resources, Steve Laviolette for his support, and all my lab mates for their helps and the amazing jobs they are doing every day in the lab. And thanks all of you for being here, attending my talk, and for your listening. Thank you very much.